Good afternoon, and thank you for joining. I'm Matt Roth at Baylor College of Medicine, and I welcome you to today's presentation. The Extracellular RNA Communication Consortium is an NIH Common Fund program which works to advance the science and research of extracellular RNA. The consortium hosts monthly presentations on a variety of research topics, and I'm pleased to introduce today's speaker, Dr. David Wong, Professor and Director of the UCLA Center for Oral, Head, and Neck Research. The title of his presentation is Saliva extracellular RNA, a new horizon of dental, oral, and craniofacial research. David?
Yes, I'm here, Matt. Thank you. So, thank you, Matt, and thank you, Dr. Valdez, for this opportunity to present our work. So, I'm here today to present to you a facet of work and research that echoes the extracellular RNA, communication consortium, and look forward to spending the time with you in the next 45 minutes or so. So, the next slide is a disclosure slide. I have a virtual company with the University of California, Los Angeles, and I'm a consultant member of entity on the screen here. So this is a project that's part of the Biomarker Development facet of the Extracellular RNA Communication Consortium, ERCC, and this is the 5th year, as many of you know, of this program, and so the particular program director to our project is listed here is Dr. Tagley and Dr. Valdez. So the project itself is schematically, represented here that this is a project to develop that encompass both the discovery and validation of saliva extracellular RNA for a disease indication, that being gastric cancer. And I'll go into the rationale of the indication selection and the pathway itself. The project centers at University of California, Los Angeles. Its clinical partner is at the Samsung Medical Center in Seoul, Korea. And we also have the opportunity to work closely with other consortium member, which is by nature part of this is ERCC in a very collegial and open manner. But there was a particular initiative to work with the DMR at Yale, particularly with Dr. Joe Wozalski in refining bioinformatics tools for microbial, containment and the value in biomarkers for gastric cancer detection. And I'm also cognizant to that while the work is primarily conducted using a population in Asia, in Korea, a validation initiative has been planted at MD Anderson with Dr. Robert Brasilia to evaluate the performance of the marker that are being developed at the Samsung Medical Center and how it performed in the U.S. population. At the same time, as many of you know, we have frequent and interaction with our leadership, program directors, through meetings and teleconferences of steering committee, as well as other venues as well. So the project in a glance is a biomarker development project that encompasses a discovery and a validation phase. We adhere to strictly, the guidelines that were set forth, through the Early Detection Research Network, prospective design, and independent, cohort in a clinical context of utility for both the discovery phase, which is designated as the first 2 years of the project, the UH2 phase for biomarker discovery. Again, this project is focused entirely on the extracellular RNA in a biofluid. In our case here is saliva, and as many of you know, saliva, while is not mainstream, it beholds the the endowment, if you will, of all these omics constituents that are present in all other bodily fluids, and this initiative is to credential the presence of these extracellular RNA constituents as well as their utility for biomarker utilization in both discovery as well as validation. So we'll go through these definitely in the later part of our talk, but is adhering to a vigorous, prospective design, having enough power, and I just want to point out here, this encompasses a discovery cohort of 100, gastric cancer individuals and 100 non-gastric cancer individuals prospectively, collecting their clinical samples saliva prior to clinical diagnosis of the lesions, and eventually advance into a validation. And I want to particularly stress the fact that we are aiming at the power of definitive validations. Okay, I see something that's popping on my screen here. So, okay, so Matt, what should I do? Approve or decline? Do you see it?
Yeah, I can see your slide. You're trying to transition it?
I see a window opening up that remote control request. Julie is requesting remote control of my shared computer. That's
decline that.
Decline that, right? Okay, all right, okay. Okay, all right, so I think we're good for now. So just be cognizant that this is a biomarker project that we are most excited about, that we have an opportunity, a very significant opportunity to develop extracellular RNA biomarker to a disease indication, gastric cancer, that has the power that at the end of the process of a definitive nature of these targets that I think in part is poising. Somehow I cannot advance. There we go. Advance these targets towards the eventual next step of regulatory evaluation. The project is anchored in Korea, so these regulatory agency is in place, as well as through our collaboration at MD Anderson. A discussion is also in place with the U.S. regulatory evaluation as well. So on that note, the Extracellular RNA Communication Consortium is an NIH Common Fund activity that encompasses a transformative agenda in evaluating and elucidating extracellular RNA in bodily fluid for a number of important areas. The basic science aspects of extracellular RNA, how they sort of convey their biological functions, and their utilities for therapeutic applications, and particularly in the context of extracellular these particular entities that embodies these extracellular RNA targets, a biomarker development initiative. That's where this project belongs. I believe there are 9 or 10 biomarker projects that's within it. There's a reference catalog initiative, and obviously there's this all-important data management core that's situated at Baylor that oversees the entire project and integrating the outcome. I think the activity today is also coordinated through DMRO as well. This is viewable on the web. This extracellular ERCC. This is one of the earlier screenshots, which in ways that also highlighted some of our early work that's being shown here. But the essence of the excitement is that RNA is no longer being sort of perceived as an intracellular, intranuclear entity. It's shed, from cells ubiquitously, either as free-form or encompassed in these particular entities, and they have the biological capabilities of conveying, from a source of origin, like a neuron here, to a distal target cells, like a colonic, epithelial cells, enter the cells and exert a biological function. So this could be within a host, or it could be in a transkingdom manner as well. So there lies the the excitement of a new paradigm that opens up for biological evaluation, as well as translational and clinical applications as well. I want to sort of share a few thoughts about the utilization of oral fluid or saliva as part of the a sort of clinical and translational landscape. So the vision of this has of saliva having this endowed capability for clinical utilization has been empowered by the scientific community, and especially being catalyzed by one of the 27 institutes, and the Dental and Craniofacial Institute, where there were really visionary initiatives to develop the salivary proteome, as well as tools that can allow clinicians for monitoring diseases, as well as health surveillance as well. So the importance of perhaps the question being asked is that why saliva perhaps hasn't reached a clinical reality is there could be multiple, but for our group here, we focus on the importance of advancing these basic translational and clinical entities in a translational frontier as well as scientific frontier. The translational frontier echoes very well into this ERCC initiative, the Biomarker Project for Gastric Cancer Detection in the context of extracellular RNA. The opportunity, the ability to discover and validate these saliva omics targets, in this case here extracellular RNA, in a definitive manner, and we'll certainly go into that. And secondarily, and equally important, is the scientific rationale why a local or more importantly a distal disease, upon its development in a host, can convey that pathognomonic extracellular RNA signature from the organopathology through vasculature, lymphatics, coming to the salivary gland and into saliva, and sort of that mechanistic underpinning is very, very important, and there lies the serendipitous, if you will, convergence with these extracellular vesicles that presents itself as a logical and compelling mechanism for advancing these information. Where does saliva come from? Saliva comes from three pairs of major salivary glands, the parotid gland on the side of our cheek, and the submandibular and sublingual gland in pairwise manner in the lower jaw. And on a daily basis, these three pairs of major salivary glands, there are minor glands as well in the lower lip. If you move your tongue to the lower lip, you feel the little bumps. Those are the minor salivary glands. But these three pairs of major salivary glands on a daily basis produce, close to a liter of host saliva that comes into our oral cavity, non-invasively, non-painfully, and non-embarrassingly. And that's it really is an abundance of a body fluid that harness that contain these omics constituents for personalized and precision medicine applications. Where does the juice come from, so to speak? This is a typical salivary gland with the acidoid cells, the ductal cells, the capillary bed that nourishes the salivary gland is really the source of the fluid, electrolytes and biomarkers, if you will, that there are active, passive, and secretory mechanisms that pull these targets into the luminal space of the salivary gland, of the apparatus, and then come into the oral cavity. Now we have the tools, now we have the knowledge base, now we have the skill set that could utilize this biofluid, and engage in vigorous biomarker development and harness these extracellular RNA targets for biomarker discovery as well as validation. So historically, prior to the extracellular, the ERCC that sort of implanted this initiative that we're all part of, my lab has been engaged in a process that's been elucidating or utilizing saliva for its molecular content, the omics content. And in 2004, we sort of began that process and profiled, a landscape of extracellular RNA in saliva. These are cell-free RNA, so it does constitute that extracellular RNA nature. We also begin to translate the utility of the salivary transcriptome for disease detection. Oral cancer is a logical first inroad, and this work was published in the same year, demonstrating that a number of these extracellular RNA that have clinical performance for detecting early stage, oral cavity cancer, and then further went on to compare the performance, if you will, of these saliva constituents compared to a seromo constituents in the same subject. So these are pair saliva and serum samples from the same subject. We're very fortunate to be working with our colleagues in head and neck here, so demonstrating the performance of these extracellular target in saliva is equally stellar, if not more in saliva. So those are the early work, and when the sort of at the same time, we also begin to evaluate the systemic oral connectivity. That if systemic disease, is fairly well known to be manifested in the oral cavity, or systemic disease, conveyed through an RNA, vehicle, if you will, being reflected in saliva. So working with James Farrow, who was here and has now moved to Yale, we sort of have a collection of almost 450, saliva samples from its endoscopic clinic prior to a histological diagnosis of these individuals. In this case here is pancreatic cancer. Okay, so and the message here is there are these genomic constituents primarily from extracellular RNA and they're being listed here that have a performance that you're seeing on the slide here. Now these are pilot study, small in number, but it really begin to credential and reveal there's a landscape of extracellular RNA. And again, we're looking at, and in this case here, in a year about maybe 8 years ago, and that concept of extracellular RNA in saliva in particular, it's not intuitive in nature, in the sense that this is hostile biofluid, which is considered by many that they may not have allow the integrity of saliva to RNA to survive in saliva. Knowing in particular, some of you may know there are actual RNases that are actually been found to be present in saliva. So this really constitutes a certain conflict in terms of this work, and at the same time through this work with James Farrow, we also begin to scientifically underpin when pancreatic cancer developed in a rodent model, and when we isolate the tumor, the serum, salivary gland, and saliva, using a high-throughput approach, looking at extracellular RNA, we were able to track these tumor-specific fingerprint, if you will, of extracellular RNA that traverses through a systemic circulation, converging the salivary gland into saliva. And what's of interest is that this particular set of work also explore a vehicle, if you will, that could be responsible for the conveyance of these molecular targets from the organopathology into the salivary gland. And the exploration at the time was these EVs, exosomes, and the exploration at the time was knocking down one of these key biogenesis member in the RAP11 gene in the biogenesis pathway, and this set of results led to a ablation or a large ablation of these target coming in saliva, supporting in our opinion for the first time that an actual vehicle can be identified that convey these pathological fingerprint or signatures from an organopathology into salivary gland. So with that in mind, that this is a bit of context that when the extracellular RNA communication program comes into the community, it was very exciting for us to sort of see that the ERCC, the Common Fund, has implanted this transformative initiative and inclusive of saliva as a biofluid that credential extracellular RNA and biomarker development. So our advances is now into the work that we actually embark upon in the past 4 years, and I'll go through them with a certain amount of being succinct, but also highlight perhaps the lessons learned, the outcome that perhaps is of value to the scientific community that credential this biofluid that has these omics constituents that could be credential for translational and clinical research, as well as a landscape of fundamental biology that could be occur within the oral cavity and beyond, and perhaps these regulatory elements are beholding those functions. The project itself is a biomarker development project, consists of a discovery phase of a prospective nature, procuring these samples, and then a validation phase, and then being cognizant that the outcome, doesn't rest upon the completion of an opportunity to definitively validate this target here. We're talking about a significant cohort of discovery and also for validation, totaling 1,500 samples, clinical samples that are prospectively procured, at Samsung Medical Center, and I'll echo that thought later on in my talk of the value of having that power to credential these targets, and importantly also is that leadership is in place to begin to advance these targets to a regulatory evaluation, both in Korea as well as in the U.S. as well. Gastric cancer, these are the clinical facts, is the fourth most common cancer. It affects particularly with high incidence in certain geographic areas of the world, South America and Asia, and early detection clearly is essential. And at Samsung Medical Center, this is where we work with leadership, in particular with Dr. Song Kim and his team. He's a gastric cancer surgeon, who's also the senior VP at Samsung Medical as well. The outcome of his body of work is due to his leadership and his clinical team that enables the project to have a procurement of 1,500 clinical samples that are prospectively procured, and I'm glad to say that we actually reached that targeted enrollment ahead of its timeline. Importantly is that in Korea that you may know that screening of gastric cancer is a national initiative. There's everyone over the age of 40 is entitled to a free endoscopy every 2 years, and the country invests over $2 billion into this screening process. So this process could be assisted by credible biomarker. It would have significant clinical and economic impact as well. So the project itself is strictly adhering to a prospective design in the context of the Samsung Medical Center in the health screen program, and this is the schematic that we strictly adhere to, and this is the distribution, if you will, of the lesion at the various clinical stages that presented as a Samsung Medical Center. The project began itself by training and also designing, a technical and bioinformatic pipelines is to use a discovery cohort of 100 gastric cancer individuals, subjects as well as controls, obtain their clinical saliva samples, pulling out these extracellular RNA and build quality small RNA library and long RNA library that that's subjected to an RNA-seq procedures, and then having that capability to analyze. So with that roadmap in place, the lessons, if you will, that were learned has been tabulated and has been categorized or cataloged, and will be available for public assessment soon. So standard operating procedure for saliva collection, processing, stabilization, and storage is in place. That is critical, and ability to standardize the clinical sample procurement, and that's in place. The initial landscaping of the extracellular RNA, having that resolution, the boundary of these different classes, if you will, of extracellular RNA has also been landscaped. A paper should be out fairly soon that actually contain these specific technological features that would allow investigators either within the consortium or outside the consortium to explore their utilization of salivary extracellular RNA for biomarker development. And I just want to sort of share with you, saliva collection is sort of, if you survey the environment, it's really a very, there are different applications, from the glorified straw to drilling into a cup. So Paul Slowey has taken the time. He actually has a commercial entity in Portland, Oregon, and this is a device that he designed for us. He called it RNA ProCell, and this is actually the apparatus that we use in our project, that has an absorbent pad with a handle that you can introduce into the side of your cheek, and in a very passive way can absorb up to 2 ml of saliva, and this apparatus is being quite clever, that this will allow the removal of cells. There's a filter here at the neck that when you express this absorbent pad in this syringe barrel here, cells will be trapped in the neck, and then the cell-free saliva would then bifurcate into 2 collection tubes. So this is sort of illustrated in the next slide here, where this is where the handle and the absorbent pad being introduced inside the cheek. You, upon about 5 minutes so it's saturated, you introduce in the syringe barrel, you express it, the cell-free saliva is being expressed and bifurcated into the 2 tubes, and you can mail this in an ambient temperature mailer, and the technology has been worked out that this transportation means of transferring a clinical sample at ambient temperature from a site of collection. It could be at a clinical site. It could be at the comfort of one's home, and then mail it through a regular mailer and to a clinical lab. Details in terms of RNA isolation, the commercially available kits that are available, they've been benchmarked and tested, and as you can see here, the New England BioLab kit in our hands has worked out to be of best performance in terms of pulling out both the small as well as long RNA. We also explore the sort of the depletion of bacterial component. We'll come back to it because as many of you know, in the oral cavity there is an abundance of microbial elements, and they're endogenous. They're there as part of the normal flora, and so their presence needs to be addressed, not only from a clinical but also from a biological point of view. We evaluated that extensively, but the final analysis is we would deplete the 16S of the microbial RNA. Actually, it would render too little amount that from a clinical sample that will allow for meaningful library constructions and downstream applications. So all of this hopefully should be coming out in a manuscript, in the next 2 months. And again, these are lessons learned, and also perhaps important outcome that we should be cognizant of as we look forward to utilizing saliva as the clinical samples for extracellular RNA. But these are time-tested, and our project entirely was foundation on it. The outcome of the first landscaping of extracellular RNA in saliva yielded a number of unique and perhaps valuable outcome that I want to share with you. If we look at the microRNA that are present in healthy people's saliva, what is being shown here is that the extracellular RNA in saliva, if you were to sort of cluster it with other available databases from serum and other body fluid and tissues, saliva cluster closely to other body fluid, plasma, CSF, and serum, which is almost sort of, it makes sense, that way. These are adjacent body fluid that may be, movement from one compartment to the next. What is interesting is we look at another class of these extracellular RNA, the Pee-wee-associated RNA. This is work by one of our senior scientists here, Dr. Fang Li, and also work with our collaborator, Dr. Grace Xiao, has discovered that for the Pee-wee RNA class saliva, is here. There are 8 samples. It cluster, instead of close to plasma, and it clusters closely to embryonic stem cell and skin. So while this is factual in this presentation, I'll come back to it at the end of my talk because I think that this is a revealing a level of information that perhaps of important biological applications, or information, and we actually explore that, and I think it opens up a new landscape of biology that we find it timely and exciting and hopefully impactful, and I'll share with you. Another class of these non-coding RNA are the circular RNA, which we through this early work we actually identify over 40 of these circular RNA in saliva, and it's present in body fluid. I think is still in its infancy, but knowing that there are this abundance of circular RNA, which amongst other potential functionality, they're docking station to microRNA. And these non-coding RNA, as we know, are regulatory in nature from microRNA to Pee-wee RNA. The question perhaps is really looking right at us is that what are these non-coding RNA? What are they regulating in this environment, in this milieu where saliva is? And the saliva doesn't stay in the oral cavity, but of course, sometimes it's spit it out, but most of the time we swallow it down to our esophagus, into the GI tract, and does that sort of present a level of biological signaling that conveys signals from a systemic origin, conversion to salivary gland, and convey itself in terms of biological functions. So that body of work actually made the inaugural issue of clinical chemistry, where they sort of present molecular targets in clinical applications. The outcome of our work is part of this extracellular RNA atlas that many of you have seen or part of. And what you're seeing here is that body fluid and clinical conditions and other utilizations. This particular schema here demonstrate that there's a good presentation of saliva extracellular RNA library. I think these are still at this time small RNA libraries. There's 243, and our laboratory, in addition to Kendall Jensen, has contributed 198 of this small RNA. Again, this came from that discovery set. The discovery set were 100 gastric cancer and 100 controls. 2 were deemed quality-wise has to be removed, so 200 were here. But there's 200 long RNA library that needs to be eventually integrated as well. Part of the the importance is the existence of the DMRR within the consortium that has built these very useful bioinformatics tools to be able to analyze the extracellular RNA for QC purpose, as well as the eventuality for biomarker development in the biomarker group, and amongst others. So this pipeline has been very wonderfully developed and time tested. But of interest is when this particular pipeline is applied to extracellular RNA in saliva, this is what we see. So this is an early chart here to demonstrate that if you look at bio, CSF, culture media, plasma, and serum, this is where saliva is, and you look at the QC of these samples, you see most of the samples hitting at 80%, which is very obviously this is what you hope for. But if you look at saliva, it registered less than 5% in terms of quality, suggesting that most of these have a presence of human sequence. Perhaps is less than satisfactory. So this is sort of our observation. But through this process here, we've been working with leadership at DMRR and actually came to a realization that the microbial component within saliva needs to be independently addressed, and one of our colleagues in the lab, Carolina Kazorbanowitz, has taken the effort to work with leadership at DMRR, and you see some of them authored here, Bob Kitchener and Mark Gershwin and Timmer and Joe Rosowski should clearly be here, but I think they just sort of truncated the authorship, has come up with an independent modification of this extra pipeline and demonstrate the importance of a number of values that Carolina highlighted on the slide here due to this high microbial content, that's an optimization of the extra pipeline and needs to be in place that really would allow the proper quality control assessment of human saliva. Perhaps another way of reading this message is that these extracellular RNA in saliva is there of a microbial as well as of a human origin, but we clearly need to have the capability to stratify and to properly identify it. And then with that in place, you can see that the advancement, if you will, using that modified bioinformatics tool, then the saliva, the QC aspects of it is at a satisfactory registration, if you will. So the project started almost 4 years ago, and one of the important outcomes from the onset is the ability to accrue, procure clinical samples for this biomarker project, because this biomarker project really encompasses an important outcome, and that is the development of extracellular RNA in saliva, number one, and secondarily, have that capability to advance these biomarkers towards a definitive validation that the outcome, if the biomarkers survive, then it have that capability of advancing towards regulatory evaluations. So for those of you that are familiar with biomarker development, that really is critical, and having that ability to do so encompass a number of diligence and discipline, prospective design, and clinical context needs to be defined, and having clinical sample with sufficient power, independent cohort of advancing these towards regulatory evaluation. So this is a slide here that we actually show in our monthly progress for about 3 months ago. What it demonstrates is that our targeted enrollment is 750 in the gastric cancer group and also in the non-gastric cancer group. And some of these are go-no-goes sort of criteria here. Is that 3 months ago in summation in the UH2 and UH3 phase, we have procured 973 in the gastric cancer group, and 818, and the summation of it exceeded the targeted enrollment, and I just want to say this with great degree of sincerity, and particularly to our colleagues at Samsung Medical Center, because this couldn't have happened without them, and for some of us who sort of been engaged in biomarker research and know how challenging this is. And I was often told that the rule of thumb is that if a clinical group tells you that this is the sort of the enrollment that they can anticipate in a certain period of time, you sort of factor in 25% of that. But this is clearly one collaboration that exceeded that and it meet our expectation, and all being procured in a prospective manner. And perhaps that really is a rationale why we sort of reach out to our colleagues over in Samsung Medical Center, and having that power is extremely important. The outcome of the discovery work led to a number of targets, which I'll show you shortly. And now we're in the validation phase of the project, where it needs to advance these targets through a successive, steps of validation. A initial step of determining the quality of these clinical samples, and then advancing through independent validation step and individual marker validation using 50 cancer and 50 control, and then a second independent validation of those targets that advance from this step here of 150, 150. This outcome here, those that validates, will be configured using logistic regression of a discriminatory panel, and that panel will be locked down and then evaluated in an independent cohort of 75 and 75. So the total number is listed here. As you can imagine, these are all RNA targets. Real-time PCR, digital PCR are being incorporated into this is performance advancement here. And I believe next slide is these are the targets that we have discovered and will be advanced to what's the validation path. What you're seeing here is 5 long human RNAs and 13 microRNAs. And what is interesting here is that while this is not initially in our plan, but working with DMRR, with Dr. Roselsky, and also through the very creative work of one of our colleagues, here, Dr. Yong Kim at UCLA, and working with the colleagues in Samsung, it's a revelation of H. pylori, and also a ethnicity-specific EBV strain. We also work with, I think is Dr. Costanza, and also Dr. Charles Repkin at NCI as well. These targets begin to emerge, and you may know that obviously H. pylori have an association with gastric cancer and EBV as well. So Carolina actually has spearheaded this with Dr. Yong Kim and discovered these bacterial targets and EBV targets are meritorious for advancing for clinical validation. I'm looking at time here. I think we have about maybe 5 minutes left, and I'll go through those fairly quickly. Now, here is sort of where we are with the project. We are being poised. Targeted enrollment is reached. Discovery is in place. Target is being advanced for validation. A glimpse into the potential eventuality of the value or utility of these targets actually could be glimpsed here at this work that we actually currently is pending revision. And this work is on extracellular RNA in saliva gastric cancer detection, again working with our colleagues at Samsung Medical Center. The next is a performance chart here that demonstrates that 12 messenger RNA, long RNA, and 6 microRNA through a rigorous development and validation process demonstrate that the RNA marker itself has an area under the curve of 0.81, which is very respectable. Demographics of these subjects, smoking history, gender, and others on its own has an area under the curve 0.69, and if you put them together, they actually have a synergistic, combination of yielding an area under the curve of 0.87. Now, this is an independent project of the Common Fund project that I just overviewed for you. What I'm trying to perhaps point out here is that the project that we're currently embarked upon is one that has a significant, power of subjects being enrolled. The clinical demographics are much more robust and in place. The saliva collection is all being calibrated. So there's a reason for us to look forward that the outcome would be advanced than this study we saw earlier here. Looking down the road, that should the outcome of this body of work translate into clinical utility, I just want to mention that there are clinical tools that actually are in place, and you may recall that there was these initiatives that the Dental Institute has spearheaded, point-of-care capabilities to a high-throughput platform using the core technology that was developed that's optimized to saliva is in place. That could be in a clinician's office, or could be in a reference lab that can address these performance or assessing risk assessment if someone is at risk for indications. Dialogue is also in place both in Korea as well as in the U.S. to begin to eventually, at the juncture where these biomarkers are validated, advance into discussion with the FDA, both here in the U.S. as well as in Korea. I just want to quickly mention two aspects. That the outcome of this work here clearly is a biomarker development project which we're excited about, and that's a good reason to anticipate that the outcome would be equally stellar, if not exceeding the performance of that microarray study that we showed to you. The biology is an element that I think is important to take note of. The extracellular RNA, particularly that subclass of pRNA that I mentioned earlier, that is very unique in the sense that it clusters closely to embryonic stem cell and skin, and of interest is a number of these pRNA are unique to saliva. So working with my colleague, Wen-Yun Shi, before he moved to Foresight, we actually have opened up a direction where these oral saliva pRNA are in crosstalk with the pathogens, microbial pathogens in the oral cavity, and seemingly elicit a mechanistic aspects of host bacteria interaction, particularly from a resistant perspective, and the manuscript is currently being reviewed on this aspect. Another biological outcome is these pRNA has a regenerative component, is being closely clustered to skin and embryonic stem cells. So when Samantha Chang, who's a DDS PhD, introduced these pRNA into a monolayer of oral keratinocytes, do a scratch mimicking a wound, and in a control dish, they will close in 5 days. And if you sort of introduce these pRNA, as you can see here, at different time points, you can really knock down these pRNA. The wound don't close. And even after 5 days, they don't close at all. So there's this ongoing interest phenomenon in the oral cavities. Oral wounds heal much faster than skin wounds, and they don't scar. And the underlying mechanism or that switch has yet to be discovered. We're not saying this is responsible for it, but a whole in the landscape of biology is being opened up due to the revelation, if you will, of these non-coding RNA. Again, the question is, there are these docking stations, circular RNA, these microRNA, pRNA, amongst others, that are present in saliva, and what could be the convey. This is a schematics here that we're looking ahead, that when an organ has a pathological entity, these exosomal entity or vesicular entity being shed convey within these disease-specific targets and converge through vasculature and lymphatics in the 3 pairs of salivary glands. And where do they go? And while they could be spit out, but most of them is being swallowed and go down our GI tract, whereas as we know, in the distal ileum, there's this mucosal lining, the pious patches, and the gut-associated lymphoid tissues that the initial discovery of these exosomes or community entity between T and B cells really present a very hypothesis testable outcome that we look forward to in terms of biological evaluation. We also look forward to there are two meetings on a national basis that this work. I will be presenting our work with our other colleagues at the ERCC in the upcoming AADR meeting, the American Association of Dental Research in Fort Lauderdale, and also in the international meeting in London, England. And I look forward to if those of you will be intended to join us and maybe engage with us in a dialogue as well. I truly want to thank the opportunity through the Common Fund making this project possible, and so looking at hindsight and how we actually integrate in the program. Our program director over the years, Dr. Tagley, Valdez, Lombo, and Kuo. The colleagues at DMR has been extraordinary in working with us. Dr. Rozalski, Roth, and Alexander, our colleagues at Samsung Medical Center, Dr. Sung Kim, Kyung Kim, and Sumi Kim. These are all gastric cancer surgeons. Actually, Kyung Kim is a pathologist. And finally, our colleagues here at UCLA, Dr. Sung Kim, Yung Kim family, who's an RNA biologist. I mentioned Dr. Carolina Urbanowitz, Shannon Rao, our statistical team, Dr. David Alishoff, and Tristan, our clinical team, and our bioinformatics, integrated biology, and also Dr. Majerim as well. So on that note, I want to thank you all for joining me, and I think we still have a few minutes to address any questions. Thank you very much.
Yeah, now we can take some questions. Thank you, David. Very nice talk. So yeah, we'll unmute everyone. Just a reminder, if you're not asking a question, if you could just put your device on mute to keep the background noise down. If you could introduce yourself and your institution, that would be great. So any questions for David?
David, this is Roger Alexander from the Pacific Northwest Research Institute.
That would be awesome.
Hi, Roger. How are you?
Two quick.
Yeah, if you could just mute your device, please. Thank you. Go ahead, Roger.
Can you, the gastric cancer, is there something specific about gastric cancer that you expect RNAs from the gastric tract to get into saliva? Or I know you looked at this a little bit earlier, but how do RNAs from systemic disease get into saliva?
Okay, so good question, Roger. So I think that the question factors into two aspects. Why gastric cancer, and perhaps, gastric cancer constitute a systemic disease, it's not an oral disease, and how does that information conveys itself into the oral cavity? So the first part is that gastric cancer was selected is not because it is 5,000 miles away, but it is a clinical group that could meet that deliverable of having 1,500 samples that are prospectively procured, because while the concept is good, but anything short of that targeted enrollment would render the validity, if you will, Roger, of these outcomes being credible. If I cannot sort of there's no other way to say this, because the credibility of the targets are really essential. So that's why we reach out to our colleagues at Samsung Medical Center, because our working history and track record, really supports that. And if the outcome is a credible one, then we can then advance it to regulatory evaluation. Now, the question in terms of how these targets are being conveyed from a distal organ into the salivary gland, the saliva. The EVs, the extracellular vesicles, seems to be a very logical vehicle, and that early work that we published when we knocked down RAP11 supports that. Now, additional work is ongoing right now to be more definitive in perhaps shutting down these exosome biogenesis. We're in the process of doing that. Others have as well. But our focus is on ablating, Roger, those particular entity in saliva, because they clearly are there, and the EV seems to be the vehicle, and sort of being able to definitively ablating their presence, I think would be the next step in sort of really connecting the systemic oral communication, Roger.
And my second question is, do you plan to do any RNA sequencing or proteomics of that wound healing assay with plus or minus knockout of those piRNAs?
So this would be RNA-seq on what entity, Roger, are you thinking about?
I'm basically wondering if you can tease out the sort of networks of systems in those cells that the piRNA is, working with to activate wound healing, or, in its absence, not to be functional.
That really is a brilliant thought. Samantha is next door, so to speak, and I think she would really welcome that thought and perhaps to follow with you. But what I can share with you is that I think that's going to be a very productive direction. What Samantha is pursuing is these PVRNA, working with Chris Falkwitz, who is now at Minnesota, identify downstream targets, Roger. Those downstream targets are the transposable element in the genome, especially of the ALU classes, and through an epigenetic, way of modulating that activity. So it's a very intriguing, axis, and I think your thought is really brilliant, and if there's any interest, we look forward to dialoguing with you, Roger.
Awesome, thanks.
Thank you very much.
Okay, David, thank you very much for your presentation, and I'm sure David would be happy to answer any questions if you contact him by email, and we will be posting the presentation on the portal.